So let's begin by talking about the test next Tuesday. You guys do know that there's a test next Tuesday, right? I like to give you this breakdown across whatever the content is for each test so it can help you guys try to budget your study time. So as you look at this, there will be 7 questions from the introductory material. That would be the history of psychology, the definition of psychology, the subfields, the biopsychosocial model, those light blue slides, right? The research method section is 14 out of 40 questions, and then almost half of them, 19 out of 40, will be on biology and behavior. So that's where you really want to spend most of your time as you're preparing for the test, and you should be preparing now. You should be continuing to do learning curve quizzes. You should be reading the textbook. You should be doing supplemental instruction sessions and so forth to get yourself ready. We will use the PAR score answer sheet. It's like a half sheet wide. It's number F289. That's the one you need to bring. It's red. Don't bring us a green one, or we're going to send you out to get the red one. We will begin at 9:30 on Tuesday. Here's the most important part about the test. If you show up more than 25 minutes late, you are not going to be given a test. You're going to get a 0. Why? Because with 500 people, and there's almost a full 500 in the section, by the time that 9:55 rolls around, that is 25 minutes after we started the test, we'll probably have 40 or 50 people who are done. And once they leave, I can't control what they do or don't do regarding that test. So I've had this rule for like a dozen years, and it works really, really well. So you have to get here on time. So if you're driving in, I don't think it's going to be rainy next Tuesday. It looks like it's going to be like 80 for like a week and a half straight. Just make sure you get here on time. Leave an hour earlier than usual, whatever the case might be. But please don't be the person showing up at like 10:15. Even if you start crying, I'm going to tell you, sorry, you're going to have a 0 on test 1. Okay? Couple other announcements, and I'll see if you guys have any other questions. My number 1 recommendation for what to do to prepare for the test would certainly be to go to SI Sessions. So you click on the supplemental instruction button here on Blackboard. There's a link through to the calendar. I've already opened up the calendar in this tab. This is the weekly set of sessions. So today, Haley's doing one at 12. Shyla is doing one at 2. Is she here? I know her brain is in the slide, so we'll look at that in a bit. Harrison, who was in the 8 o'clock class, is having one at 4:30. I would recommend not saying to yourself, well, what I'm going to do is next week on Monday, right before the test on Tuesday, I'm going to go to Tim's last session and just try to cram everything in that last hour or 2 of the day there. Not a good idea. In fact, this brings up the tip of the week that I'm going to play here from one of our SI leaders who's here this morning. So this is from Tessa and what she recommends for preparing for the test. Tip of the week here.
Hi, my name is Tessa. I'm a supplemental instruction leader for spring 2016. My tip of the week is that students who come to sessions early on in the semester often do better than students who come to just the review session. So I invite you to click the button on Blackboard, attend an SI session, and start learning with me.
Awesome. And she didn't get attacked by a turtle down there by the pond, so that's the best part there. I do want to show you one other thing because I had a couple of questions over the weekend. It's hard to remember passwords and type them in while you're talking. About quizzes and other stuff here within Launchpad. So let me just jump out to your section just so it says 9:30 on it. In Launchpad, what's the only thing that is assigned to you that's worth points? The learning curve quizzes. You can do a whole bunch of other things, and I'd actually recommend that you do other quizzes and other activities that are built in there, but the only thing that's worth points is the learning curve quizzes, which you can access again one last time in one of 2 ways. Click on the calendar over here, and when you do, you'll see each of the learning curves sitting there. Or if you don't like doing it that way, you like to do it within the chapter. Maybe you're in the bio chapter and you're working your way through section 2A. There's the learning curve 2A. You go in and access it that way. Okay? That's the only thing that is worth points. There are 10 points worth of these per chapter. There are 13 chapters, That means you could get 130 points. Remember, the maximum points for the learning curve quizzes is 120. So even if you miss one, as long as you do all the rest, you're good to go and you'll get all those points. One final thing on these quizzes. The points here will land and remain in the Launchpad gradebook, and they won't appear in Blackboard until the very end of the semester, okay? It's not integrated tightly with Blackboard in the way that I would want it to be. So what I do at the end of the semester is I pull these grades down into Excel. I add up all your quizzes that you've completed. I get you one grand total, 110, 120, whatever the case might be, and then I go and add that into a new column, a new grade in the Blackboard gradebook. So for right now, if you want to know what your point total is here, you do that by looking within Launchpad. They won't appear in the Blackboard grades. Does that make sense? It's a separate thing for right now. All right, any other housekeeping questions or things you're worried about or don't know how to do? Bless you. All right. Also, just a reminder, especially because I feel like we had more than a handful of people add this class fairly late. Everything that we've done, let me make this bigger. Everything that we've done is recorded. All right. So our Tuesday lecture recordings are right here. If you click on the button, there'll be a new one there today that will say 2/9/16. Biology and Behavior Part 2. So the Tuesday sessions are on the Tuesday lecture recordings button. The Thursday online sessions are on the Thursday online recordings button. You've got 3 of those now. This was the first one. This was the second one, and the biology was the one we did this past Thursday. When you click on any of those, you'll be able to either view it as an MP4 or as a Collaborate session. Either way is fine. The MP4 you can download and stick it on your favorite device, and you can listen to the lecture as you're at the gym or riding on the trolley or whatever the case might be. All right? Any other questions on any of that? We good to go? All right. What I want to do is circle back to a couple of the high points from what we did online on Thursday. Oh, shit, I already got it running. Where is it? Forgot to start up a session with you guys, so let me do that. All right. I need you guys to settle in and quiet down because I'm not going to talk over noise and all that. All right, good. Thank you. So, when we were getting rolling here in the online session on, why is this not going? Okay, now we're good. On Thursday, we began with a review of these tools of the neuroscientist. So I'm going to spend about 10 minutes hitting some of the high points from Thursday and then launch into the new material. You should, for all 5 of these, be able to talk about what it does, what kind of information we get from it, and what its limitations might be. What does it not tell you? So, for example, with an EEG, you should be able to say, well, it tells me about electrical activity. It doesn't give me a brain image, and it actually doesn't tell me about anything that's deeper in the brain, that is more in the center of your head, than the overlying cerebral cortex. Remember, the cortex from the postmortem brain specimen is all this bumpy stuff on the outside shell. Okay? So if your question is about something at the level of the cortex and you don't want a brain image, an EEG might be a tool for you. If you want a nice picture of the hippocampus, you're not getting that with an EEG. The PET scan, we said, gives us information about brain functioning. Really nice look here at what areas are more active or less active as you're doing different activities. So you can see here as you're processing language, if you contrast that with the baseline, we see more sort of reds and oranges on that left side of the brain. Those active areas light up red and orange. What we're doing is tracking this radioactively tagged glucose. So where is the fuel being burned up? The logic is then that that's the part of the brain that's active when you're doing something maybe with language or with music or whatever the case might be. So good information about function. Not a beautiful picture here about structure, right? We've got to look down into the brain from above here. This is called a horizontal view. We're seeing down into it, but we don't see neatly what's going on structurally. Look at this is a beautiful picture of the brain. So Here's your brain structure strategy. This is the tool to use if you want to get a good look at the overall brain structure, but maybe not necessarily functional information. So this is what's called a sagittal view, where we basically tell the computer to split the brain down the middle in the image that you provide here. We see the overlying cortex. That's really nice. This is the corpus callosum here that we're going to be talking about today. Here are some of the midbrain structures like the thalamus, down on into things like the medulla. There's the cerebellum on the back side of the brain. Okay, really nice picture of brain structure. If your research question or your clinical question is about asymmetries in the brain, is one side of the brain bigger than we would expect it to be versus the other side, maybe that's your research, then MRI pictures of brain structures would be the way to go. If you want to have your cake and eat it too, then what you want to do here is an fMRI. And the reason I say have your cake and eat it too is that it lets you look at both brain structure and brain function. And this is, I mentioned on Thursday, an fMRI that one of your SI leaders actually emailed me last week, and she said, it's okay if you show my brain to all the students. I'm like, okay. So this is Shyla's brain. She was in an fMRI study and actually still is up at UCSD. Look at that really nice picture of the corpus callosum here, some of the midbrain structures. We've got the overlying cortex, and we're seeing some areas active as she's doing whatever the task was when that was being done. Okay? So again, know something about what each one does or doesn't do, when you might use them, when you maybe might not use them. We had this in here. Let me just make this big to make this a little bit more visible there. We've got everything covered there in infographic 2.3, other than we didn't talk about the CAT scan. So when we're talking about the test in the 8 o'clock class, somebody raised a hand and said, do you ask questions that come just from the reading in the textbook? And that's material that you didn't necessarily mention in lecture. And I said, yes, I do. That's the bad news. The good news is there's usually only like 1 or 2 or 3 of those. So for example, we talked about EEGs, PET scans, fMRIs, and MRIs. We didn't talk about CAT scans. That's maybe a good one to read a little bit about in the textbook. So those, what I call textbook-only items, are related to things that we talked about. They're not questions that are supposed to be like a scavenger hunt for you to be like, oh, where is he going to ask a question in this chapter? Okay? It should be connected in with something that we did talk about. So, for example, in part 2 of the course, we're going to talk about sensation, and we're going to go through vision and hearing really thoroughly. And then I'm going to tell you to read about those other senses in the textbook. So I might ask a question about that kind of stuff for test 2, where it's related to what we did talk about, but I didn't have time to cover all of it. Okay, that's the way I would think about those kinds of questions. All right, I do want to use this clicker question to get us rolling here this morning. So for those of you who were there live Thursday or viewed the recording, this is review. If you weren't, well, you should be doing that. So take a look at this. Sophie, who has a bad fall, she gets a brain imaging technique. Things look to be intact, but they're not sure about brain functioning. What did they do with her? All right, last 15 seconds or so here. Let's take a look. Oh, that's pretty good. Almost 90% of you saying MRI. How'd you know it was MRI? What did we have? Brain image, and we had information about structure, but not what? But not function. Okay, really good. So you'll see, right? That's an example. It's an application. It's not about some definition of one of these tools. So that's what I want you guys to be able to do. You're going to have a lot of test questions that look like that, and not so many that are strict definitions of terms. Here's the deal. I'm not going to run through all the major parts of the brain again. There are about a half dozen or more that you should be familiar with. Beginning with medulla pons, we talked about the thalamus. Cerebellum So the cerebellum has to do with muscle movement, coordination, and balance. They have a picture of a soccer player, dancer, figure skater, gymnast, whatever the case might be. We mentioned the cerebellum is one of the first parts of the brain negatively affected by alcohol intake. So if the cerebellum starts not working as well, oh, that explains why the drunk people on cops always fall down so much, right? Okay, so now I get that. There's the connection. Then we did the 3 parts of the limbic system here. We said if you want a mnemonic, it spells HA, H-A-H. So it's hypothalamus, amygdala, and hippocampus. You can review what I said about those in the recording in terms of the aspects of functioning they're involved with. I'm going to skip that one because it was too easy. We ran through then a review of what we had already done, and we turned our attention then to the overlying cortex, this bumpy outer shell that surrounds the midbrain and hindbrain structures. And then we ran through a review of these 4 lobes of the cortex. This is probably 2 or 3 questions right here in terms of what the lobes of the cortex do. So we spent the most time on the frontal lobe and said the frontal lobe was involved with things like, well, intelligence, abstract thinking, logical reasoning. Number 2, we said the frontal lobe helps control movement. It's got a strip here at the back of the frontal lobe that we said is called the primary motor cortex. It's light blue here, left hemisphere and then right hemisphere. 2 aspects of that motor strip that we talked about. Number 1 was contralateral control. So when I wave my right arm, that's controlled by the left hemisphere of my brain. When I wave my left arm, that's controlled by the right hemisphere of my brain. That's called contralateral control. And then this weird picture surrounding that slice of the cortex is meant to depict this proportionality principle that we talked about. So proportionally more brain volume dedicated to parts of our body where we could do more complex movements. And by the way, both of these things apply to the somatosensory strip, which is at the front of the parietal lobe here, this lime green strip. That's about touch, contralateral control. So if I rub the palm of your right hand, you're going to see that lighting up in the left hemisphere and vice versa with your left hand. So contralateral control for touch at the front of the parietal lobe. And then again, proportionally more brain space dedicated to parts of the body now that are more sensitive to touch. So we mentioned those things regarding the motor strip here and the somatosensory strip right adjacent to it at the front of the parietal lobe. The other frontal lobe aspects we talked about, besides intelligence and abstract reasoning, and then we talked about movement, we talked about executive control, laying out and then executing a plan. I want to do these 4 steps to complete this task today. The frontal lobe helps you to do that. And then the final thing that we mentioned was inhibition of impulses, right? And there was somebody in the online session who was like, this explains a lot when I mentioned that the inhibition of impulses part of the brain doesn't fully develop until you're past your teenage years, right? And so when you look back and think, oh, when I was 15 or 16 and I did that goofy thing and I didn't think about all the consequences, the reason why is the part of the brain that makes you think, we should slow down here, tap on the brakes, right? That's what the frontal lobe does for you. It helps you inhibit those impulses. It's not fully there yet when you're 14, 15, 16 years old. It continues to develop till about age 25 or so. Parietal lobe is sense of touch. Occipital lobe was vision. Temporal lobe was hearing. We talked about those guys on Thursday as well. Any questions about the major lobes of the cortex at all? Okay. Like I said, 2 or 3 questions probably right there. We used Phineas Gage to talk about the frontal lobe. Again, inhibition. We said what happened was after Gage's accident here where this tamping iron was blown through his skull, he became really disinhibited. He was like a guy that didn't have any brakes. There was not much editing of his thought processes. He would just say or do whatever came to mind. Visual cortex, temporal lobe, visual cortex back here in the occipital lobe. And then the final thing we did on Thursday online was a brief review of this stuff about language. We're going to come back to this in the section on development when we talk about language development in babies. There are 2 independent areas. Broca's area would be lighting up for me right now because it's involved with speech production. It lies adjacent to that motor strip at the back of the frontal lobe right here. Helps you move your mouth, your lips, your tongue, your vocal cords to produce speech. And then Wernicke's area would be active for you guys as you're listening and hopefully comprehending what I'm saying, right? The top part here of the temporal lobe, top rear part, helps you to comprehend what's being said to you. Okay, so we mentioned those as well. All right, let's finish this review with this other clicker question, and then we'll launch into the new stuff in just a minute. So if you wanted to get a cat to twitch its whiskers, that is to move its whiskers, how would you help that movement to occur? Which lobe of the cortex would help you get to that goal? All right, last few seconds on this one. This question basically comes down to where's the motor strip, right? Where's movement control? Do you guys remember where that is? Well, 70% of you do. It's the frontal lobe, right? So at the back of the frontal lobe, we have that primary motor cortex. Right here, this blue strip in front of the adjacent green strip, which would be the somatosensory strip. So movement is frontal lobe, so the answer choice there is B. Okay? All right, let's talk about the next major part of the brain here. We're going to spend about 20 minutes or so taking a look at the corpus callosum. So if you're having a snack right now, we got a nice juicy brain to look at while you're doing that. The corpus callosum in a sagittal view here, where we split the brain into left and right halves, looks like this. I like the view on the right even better because it makes really clear what this structure is doing. This structure called the corpus callosum, we're going to say, is basically a bridge between your two hemispheres. Okay? It looks to me like just a whole bunch of like rubber bands, doesn't it? Kind of connecting the two sides. And this is important, by the way, because there are certainly going to be occasions where you're going to want to share information back and forth between left and right hemispheres. Okay? I like to use the analogy of a drag and drop scenario. So there might be information that for some reason landed in your right hemisphere that you're going to want to share with the left hemisphere. Well, the good news is you've got a way to do that. You've got this bridge, this network of fibers that lets you slide that information across the corpus callosum. Okay? This is a horizontal view down into the brain. This is obviously a postmortem brain specimen, and they've sliced off the top layer of the cortex here so we can get a good look at it. I also want to mention here, both on the iOS side and on the Android side, for your cell phones, there's a really awesome free app that's called 3D Brain. It's cool. Don't do it right now. I always wonder, as soon as I say it, you're all going to be sitting there the rest of the class. You can look at individual structures. You can rotate it in 3 dimensions. The one on the corpus callosum is so cool because you can kind of rotate it forward towards you and really see kind of the curvature of the structure as it works its way through, and you're rotating from the front of the brain to the back of the brain. So when you click on each of the structures, it will tell you a little bit about what it does. It has links to like research articles about these topics. So it's more than you need, but if you want some visuals to go along with this and be able to rotate in 3 dimensions, it's really, really cool. All right, so 2 things about the corpus callosum. They're basically related ideas here. That it's this network of fibers that lets you exchange information between the 2 hemispheres. So let's say in terms of movement, that, okay, we know that the right hemisphere controls the left side of the body and the left hemisphere controls the right side of the body. What if I wanted to coordinate a movement where both arms went up at the same time and met up here, and then they went back down and landed at my sides at the same time? Okay, jumping jacks, right? In order to do jumping jacks correctly, that is synchronize the movements, we need the right motor strip to converse with and communicate with the left hemisphere. And we need the left hemisphere motor strip to share information with the right hemisphere. And we do that over this bridge that lets us do this dragging and dropping back and forth. Okay? So I would say the key word there is bridge. In 99.9999% of the cases, this is an automatic kind of process. We don't think about it. We're doing this all the time. We're going to talk in about 3 minutes about cases where there is no bridge, there is no corpus callosum. Okay? And when it's not present, it makes things get really, really interesting because you can't share information back and forth. We basically have two separate independent hemispheres, a right hemisphere that doesn't talk to the left hemisphere and a left hemisphere that doesn't talk to the right hemisphere. And when that goes on, things get really, really interesting. We'll get to that in a minute. Before we get to that, let's describe typical cases, right? So, especially in this part of this lecture in particular, I run up against this. You have to sort of tell the general story for most people. So the general story for most people is as follows regarding how the hemispheres work in conjunction with each other. What we've got here on this slide is information being presented to your left visual field and to your right visual field. The way that we typically do these tasks is we have you focus your vision, your attention on a focal point there. So that X there is going to be where I'm going to ask you to look, right? Look at that point, and that's your focal point. Well, with respect to that focal point, you have a left visual field, which is stuff that's on the left side of your environment, and you have a right visual field, which is the stuff that's on the right side of your environment. So as I stand here like this, right in the middle and the back wall is where the projector is. So you guys over here are in my left visual field, and you guys over here in my right visual field. Notice that I'm talking about the world and not left eye and right eye, okay? Because it turns out if you look at the coloring here, the left visual field information actually goes to both eyes, and the right visual field information goes to both eyes. So we're not talking about left eye versus right eye. What we're talking about is the world out there as you look at it in terms of these left visual fields and right visual fields. Here's what happens. The left visual field information ultimately gets routed to and dumped to the right hemisphere. So the left side of your visual world lands in the right hemisphere of your brain. Where does the right visual field go? Well, it comes over here to the left hemisphere. Okay? Now, we're going to talk again. Generally speaking, most of the time, and more true for right-handers than left-handers, and 90% of the population is right-handed, by the way, language and speech are specialized in the left hemisphere. Again, in most cases, this is the case, right? So let's pretend that speech/language here is this left hemisphere activity, and we already saw that again this morning when we did a little review and looked at the PET scan, right? We saw more reds and oranges on the left side when it was a language stimulus, okay? Let's pretend that language is like this. Let's pretend that the language capacities that you have in your left hemisphere are kind of like a giant Post-it note system that let you label all the stuff in your world. Okay? They let you, I see a giant skateboard back there, so I've got a way to label that with language, skateboard, and I see a baseball cap, and I see a light, and all these things, language lets me label that stuff. Well, the way that this works is that your right visual field ends up right where the language center is. So that's easy, right? The things over here are even easier for me to label than the things over here in my left visual field. But for me and for almost all of you, probably all of you, your left visual field, which gets routed to your right hemisphere, to label that information with language, what do we have to do with the information about the pencil? Where do we have to get it to say that I saw a pencil? It's got to move from the right hemisphere where? To the left hemisphere. We've got to get it over here. How does it get over there? What does it venture across? That corpus callosum right in the middle there. We drag and drop it, we slide it across, and instantaneously we say, okay, well, on the left side there, as I looked at the X, I saw a pencil. How did that happen? Left visual field pencil goes to right hemisphere. Right hemisphere gets slid across the corpus callosum, and we label it with language, and we're good to go. Again, that's 99.999% of the cases. In the 0.001% of the cases, we have individuals who have a severed corpus callosum. Okay? That's not an accident. It's not like, oh, dude, I fell down the steps and I severed my corpus callosum. Okay? It's a surgery that's done as a treatment of last resort, a Hail Mary pass kind of surgery for folks who have really severe and treatment-resistant epilepsy. So they have a bad seizure disorder where what happens is effectively what goes on with this model of epilepsy is you get like freezing up of messages that are going back and forth across the corpus callosum. You know when your computer gives you like the rainbow circle of death on the Mac side? You're like, oh shit, right? That's kind of what happens with epilepsy across this corpus callosum. So it turns out, I don't know who did this for the first time, it turns out that when you cut that, then you don't get this locking down and you get a significant decrease in the number of seizures, okay? But when you take away the bridge, what are you not going to be able to do with the information now about the pencil? You can't slide it over. There's no way to get it over there. So it ends up having some really interesting consequences for what you can and can't do as what's called a split brain patient. So a split brain patient is an individual who's had that corpus callosum severed. Yeah.
Would that cause problems with depth perception, like people who are blind in one eye?
So could it potentially affect things like depth perception? It could affect a whole bunch of things. Depth perception may be one of them. What I don't want you to get locked down in is the depth perception thing about one eye. Remember, what we're talking about is visual fields out there, not left eye and right eye. So don't mix it up with left eye and right eye. So we got, again, let's imagine we have a split brain patient, really bad epilepsy. We do this, slice down the middle of the brain here. We basically detach and separate the two hemispheres from each other. They're two independent hemispheres. I know they did this in an episode of House in like season 7 or whenever the hell it was, and it was pretty cool. So let's take a look here. Here's a split brain patient who, conveniently for us, has a transparent skull. We can see down into his head, and we can see that he doesn't have a corpus callosum. I want to remind us that we're going to assume he's right-handed for us, that language is specialized on that left side of the brain. It does the labeling for us. That's great. Okay. We have him focus on this dot in the middle of the screen in front of him. And briefly, what we do is flash a picture in front of him. We flash a picture of a key on the left side, which is his left visual field. And we flash a picture of a ring on the right side, which is his right visual field. Tell me what goes where. Where does the information about the key land? The right side, right. Why does that happen? Because we've got left visual field being routed to the right hemisphere. Okay. It actually has to do with this optic chiasm thing back here that I'm not even going to get involved with, but it has something to do with this crossover right in here. You don't need to know that. Okay. The ring, the right visual field information gets routed to which hemisphere? Left hemisphere. Okay. So when we ask him, what did you see on the screen in front of you? What it says here is he says, I saw a ring. Okay. Okay, so why is he able to do that? Well, the reason he's able to do that is because the ring from the right visual field was routed to the left hemisphere. What capacity do we have over here? Language. So we can label that. We got a post-it note for that, and we say, that's a ring. That's what I saw. Now, here's where it gets interesting. If we ask him, what was flashed on the left side of the screen? Can he tell me that he saw a key on the left side of the screen? No, he can't. Why not? Something wrong with his vision? How would you explain his inability to say, oh, I saw a key on the left side of the screen? Can somebody explain why he can't do it? I don't expect you to know this yet, but what do you think?
Because the language production is on the right side.
So language production is in the left hemisphere. But the key landed where?
It would be on his right side.
And he can't slide it over to where language is, right? So that's exactly it. Language is a left hemisphere capacity. Again, just for sake of simplicity and intro, we'll say that that's where all the language is. The key from the left visual field goes to the right hemisphere. Because he can't slide it over to where the post-it notes are, he can't label with language that object from his left visual field. Okay? Does everybody see why he's limited in that way? Here's where it gets really interesting. And I think it starts to turn into like a circus act. We say, okay, so you can't tell us what was on the left side of the screen. No, I think the analogous situation for us would be like, when you can't think of that actor or actress's name. Oh, what was that dude's name? Like, it's there, but you can't get it. That's kind of the feeling here, I think, for the left visual field. Like, I know there was something there. I can't think of it. He can't label it. But you say, let's just try something here. Use your left hand to reach behind the screen there and pick up the item that was flashed on the left side of the screen. Remember, he couldn't say with words that it was a key. What's he able to do with his left hand? Picks up the key. Like, what? How can he do that? What do you think that's about?
He still saw it, he just doesn't know the word for it.
He saw it, he doesn't know the word for it, he doesn't have the word for it, but why does his left hand, what? How can his left hand do that? Tell me about left hand. Controlled by the right hemisphere. What does the right hemisphere have? It's got key, so he can actually use his left hand to pick up a key which he could not tell you what it was with words. What? That's crazy, right? I can't say key, but I can use my left hand to pick it up because effectively he's got these two separate and independent brains now, right? And the right side, the right hemisphere has got the ring information, so we can use the left hand to pick it up because that right hemisphere controls left side. Yeah, way in the back.
If you took the key and you moved it to the opposite.
So if we move the key to his right visual field, or if we let him do whatever he wants, if he turns his head, what do you think he's going to be able to do now? Is he going to be able to say key if we flip the key over to here? Yeah. And so I think, if we let him do whatever he wants, I haven't ever worked with a split brain patient, but in terms of being able to label stuff with language, I'm sorry to keep turning my back to you, but it makes sense when we're doing this, right? If I want to be able to label that key with language, and it's over here, what do I have to do as the split brain patient to get it where I need it to be? I'm going to be turning my head, and now I got it. It's a key. Like, I don't know what it is. It's a key. Right? Just with a little turn of the head, what you're doing is you're getting this right visual field information dumped over into the left hemisphere. I was only half joking last week. We were sitting at home, and I have a certain place where I usually sit when we watch TV, and it's at a spot where the TV is a little bit to my right. And my wife sat there, and she goes, "It's weird sitting over here." I'm like, I can't sit anywhere else because I want this information in my right visual field just a little bit so it gets where the words are, so I can talk about things. And when I yell at the referee or whatever, I got the words there, right? Because it's a little, I have an intact corpus callosum, but it just feels a little bit better to me. When I go to lectures or classes, I like to sit over here. I like to get the professor or the speaker over to my right. It's a little bit weird, I know, but that's given what we're learning here. Which hand could this guy use to pick up the ring? If we had a ring behind the screen, which hand could he use to pick it up and why? Who's got it? Don't all jump at once because it's really intimidating. Yeah. He could use his right hand, and why could he use his right hand to pick up the ring? The right hand is controlled by left hemisphere, and the left hemisphere has what information? It's got the information about the ring. Perfect. Okay? Let's do one more example here, and I'll see what questions you guys got. We're going to briefly flash a picture of an apple on the right side of the screen here. She says, I see an apple because right visual field goes to left hemisphere. Left hemisphere's got language. Cool. Briefly flash a picture of the apple on the left side. I don't see anything. Or she might say, I can't think of the word. Something like that. Because left visual field goes to right hemisphere. She's a split brain patient, so she can't slide it over to get it labeled with language in that left hemisphere. Okay. Flash it again to the left visual field, goes to the right hemisphere. She can use her left hand to pick it up correctly because, again, left visual field goes to right hemisphere, and then right hemisphere controls the left hand. Okay? Let me stop there for a second. Questions about that?
So, if she, on the third picture,
Over here?
if she asked to pick up the apple with her, what she saw with her right hand, would she not be able to do it?
Great question. The question is, could she be able to pick up the apple with her right hand? Yes or no? No, because the right hand is controlled by left hemisphere, and the left hemisphere doesn't have the apple information. Okay? So here's the deal. I'm definitely going to ask you a test question about this next week. So when you're taking the test, if you want to be sitting there and be like left, and no, you're not doing the Macarena or anything. It's just right hemisphere. You guys are so young that you don't even remember the Macarena. Okay, right hemisphere goes to left. Right visual field goes to left hemisphere. Left visual field goes to right hemisphere. Yeah. I got to come back closer to you. You're soft spoken and everybody's coughing.
In the middle picture where she
Right, so when she says, I don't see it,
Does she like literally can't see it, or she can see it but can't?
great question, yeah. I don't think she would really say, I don't see anything. I think she more likely would say, there's something there, but I can't think of the word. And again, this is flashed really quickly, and if we let her do anything she wanted, like, if we say to her, well, do something that will help you be able to tell us what that is, probably what she's going to do is turn a little bit to the left, get it in the right visual field where it will dump to the left side where we've got language. Good. Other questions? Yeah.
Is memory on the left side, so like that's why she can't remember
Yeah.
the address that it was?
Great question. Where's memory involved in all this? Memory's on both sides. So we've got the hippocampus actually is in both hemispheres. And somebody asked after the first class, well, couldn't you remember that you had seen something? Yeah, certainly, yeah. That's part of it. And that's why you might be flashing any of these different objects so that you're not getting things right just because you remember what was going on there. Okay? All right, let me give you a question that's just like one that I might give you next Tuesday about this split brain stuff to make sure you got it together. Try it yourself. Don't talk to your neighbor, because if you guys don't do well enough, I'm going to let you talk to your neighbor anyway. So a picture of a comb is briefly flashed in the left visual field of a split brain patient. What is she going to be able to do? I can watch the percentage in the different answers on my screen here. So I'm keeping an eye on it. And with this question in particular, the longer the question is open, the smaller the percentage correct is, because it's a challenging question. So if you're struggling with it, don't worry. No problem. All right, let's take a look at what we've got. Okay, about half of you guys think she can use her left hand to draw a picture of the comb, and about a third of you think, well, actually the opposite, that she could use her right hand to draw a picture of the comb. So here's what I'm going to do. This is one of these, what I call peer instruction questions. It means you get a chance to talk to your neighbor, so I'm going to flip the lights on. You're going to discuss it and explain to them left and right, visual field, hemispheres, and all that. Come up with the right answer, and then I'm going to ask it again. I'm going to walk around and listen in to your conversations. All right. Somebody in the back said contralateral, and I got really excited. Okay. So let's do it a second time, and then I'll show you guys the results here. So let's just fire it up again. And almost invariably, what's going to happen is more people are going to get it right the second time. It's not like bad information flows through. This is like a Wikipedia thing, right? The correct information, the correct answers flow through. So we'll let this run all the way through, see what happens here. If you get it right the second time and you want to high-five your neighbor because they helped you get 2 points instead of 1, that is totally fine. There have been occasions where people were chest bumping each other in the back of the course. whatever you got to do here. All right. Last 20 seconds here. That was good. And what I like about these questions is it changes what's going on in the classroom, so it's not just me yammering at you for 75 minutes, because I've already been doing this now for more than 100 minutes. I like to be able to shut up and let you guys talk to each other and figure it out. Nice. That's good. He's high-fiving the guy behind him. He doesn't even know him. All right, so you guys really care about like every point. So the explanation that I heard in the back from these guys was really, really good. So I'm going to put you on the spot and ask you to walk me through what you said. So it begins with, where is the image in the blue shirt? I'm sorry, I didn't get your name. Leah, okay. So the image is where? Left visual field. Where does that get routed in the brain? Right hemisphere. And then here's where she said contralateral control. I got really excited. Right? And you said contralateral control, which means what? The left side of the body, so you went with A. Okay? And they were going back and forth with A and D in the back there. By the way, test-taking strategy. Look at choice A and look at choice D. What are they? They're opposites. Generally speaking, probably 99% of the time, when two answer choices on a multiple choice question are opposites, what's going to be true? One of them is going to be right, right? One says up and the other says down. Oh, well, it's probably one or the other then, right? So when you get to, and you'll get probably one or two of these per test, where there are two things that are saying the opposite, just get rid of the other stuff. Forget about B and C, because these two are saying opposite things, okay? And then use, what you can from there to try to, at least at that point you're down to a 50-50 chance versus 1 in 4. Questions or any confusion about that at all? Cool. I really like that little segment on split brain. I think it's fascinating stuff. Yeah.
How do you split a brain?
How do you split it? They do brain surgery. So without getting too graphic, they're going to open up your skull and they're going to have a really sharp knife. Not like a butter knife or anything. I mean, it's got to be sharp, right? I think it's called a scalpel, and then they're going to slice and dice. It's probably going to make noise. Okay, I'll stop. All right, yeah. All right. We're moving on. So let me get to this segment here. We got about a half hour to go, and we got a fair little chunk of material to get through. I want to talk about hemispheric specialization or brain lateralization, neither of which is like an easy term. It basically is going to be a little segment on what are the kinds of things that look like they're, generally speaking, more specialized in the left hemisphere, and what are the kinds of activities that seem to be more specialized in the right hemisphere. Here's what I'm not saying. I am not saying that you are left-brained or right-brained. Seriously, you're giggling because this is one of my things, right? Don't ever post something that says, oh, I'm left-brained. Oh, I'm really right-brained. You're both sides-brained. If you were only one or the other, you would be dead, right? So you might be better at things that the left hemisphere is specialized for, or you might be better at things that the right hemisphere is specialized for, but you're not one or the other. That's almost as bad as the 10% thing. Oh, we only use 10% of our brain. Well, just saying that means you're using less than 10% of your brain, right? That's also a ludicrous statement. Again, what we're saying is, generally speaking, and by the way, this stuff is more true for right-handers again than left-handers. I know the left-handers are like, I'm being totally discriminated against this whole lecture. It's because 90% of the population is right-handed, and I'll tell you what the differences are at the very end here about how this is a little bit different for lefties. Left hemisphere, language and speech, we've beaten that into the ground. Okay, if you can't remember that next Tuesday, you're going to be in big trouble. Okay, language, left hemisphere, and speech. Verbal memory. So remembering what your brother said at his, best friend's wedding when he gave the toast and you were there. Okay, that's a verbal memory. The left hemisphere also tends to be better, generally speaking, at analytical processing where it's like a checklist. I'm going to do step 1, step 2, step 3, and step 4. Sounds a little bit like that executive control thing that we're talking about with the frontal lobe, right? So when there's a step-by-step set of procedures to follow, That's a left hemisphere activity. But I just thought of this standing here talking to you guys. Like building a complicated Lego model shows you why it's not one or the other side of the brain. Because the step-by-step part of Lego, doing page 1 and then page 2, is a left hemisphere activity. The visual spatial stuff of putting things the right way, that's more of a right hemisphere. So you're doing all of this, right? It's not one or the other. That actually is a pretty good demonstration of what I'm trying to drive home, right? Not one or the other, you're doing both. So right hemisphere is those spatial abilities, right? Does this piece go this way or that way? Spatial abilities would be you're walking through the mall, you look at the directory, you're trying to find where the Apple store is, and it says, you are here. And you're like creeped out for like just 5 seconds or so. How do they know where I am? It's really disturbing. And is the Apple store to the right? Is it in front of me, behind me? Where is it, right? Those visual spatial abilities. Music, we saw that in the PET scan slide. Visual memory. Not what your brother said, but what color tux he was wearing or what color dresses the bridesmaids were wearing at that wedding. Visual kinds of memory. And then the right hemisphere is general. It's sort of abstracting the overall picture and telling sort of the story of the whole rather than wanting to go at it in a piece by piece, part by part way. Does that make sense? So left hemisphere wants to kind of break it down one item at a time. Right hemisphere is going to be better at looking at what I'm calling here these global patterns. What is the overall picture here? What is the kind of overall summary, I guess is another way to say that. Okay? So looking at data, looking at a picture, looking at a painting, and sort of abstracting the big ideas rather than looking at the individual pieces and the brushstrokes that you could see. Maybe that's a nice way to think about that difference, right? The left hemisphere is going to be better at looking at individual brushstrokes, whereas the right hemisphere is going to be better at looking at the overall picture here. Okay? For the lefties, the major thing that's different is that the left hemisphere language specialization tends to be a little more balanced in left-handers. So it's a little less of like only the left side's got post-it notes for language. In left-handers, that's a little bit more balanced. Okay, that's the major thing that you could talk about in terms of lateralization for lefties versus righties. And then you get cases that are weird. I don't know how many of you guys are like this, but my youngest, I don't know where I'd put him in terms of being left-handed or right-handed. And there's probably 5 or 6 of you guys like this in here. This is a kid who writes right-handed, but cuts with scissors left-handed. For the right-handers, like if you cut with scissors with your left hand, what would you do? You'd probably cut your damn finger off, right? Okay, like I can't even, what? Right? This is a kid who throws right but bats left for baseball. Which actually, that's awesome. That's what you want to do. That gets you a big advantage. When he plays soccer, he plays left-footed. It's this weird combination. And I think actually the deal with him is right before he got discharged. It's not funny, but right before he got discharged after he was born, they discovered he had a broken right collarbone. And so he got like cinched up, not only like the baby burrito, but like his arm cinched up, and he had to do like baby PE, PT, physical therapy. And I think he's a kid who probably was going to be right-handed in terms of the nature piece, and like, oh shit, you can't use that right hand. So he started just slapping around at things with the left hand, maybe more than he otherwise would have. How many of you guys are like that, where you do like this weird kind of mix of both? Like he bowls left-handed, but throws right-handed. So there's about a half dozen. So that's what I'm saying. With an intro class, like, here's what's generally true, but there's always going to be these individual cases that are a little bit different and are sort of, exceptions to some of those general rules. So I just think he's amphibious. He's not neither. I know it's not amphibious, but there it is. All right. Let's talk about how all this is organized here. We got 20 minutes, and I got about 3 main points I want to drive home. Everything that we've been doing so far, guys, falls under the heading of the central nervous system, the CNS. The CNS is comprised of your brain and spinal cord. So all the parts of the brain, medulla pons, cerebellum, thalamus, hypothalamus, all of that is central nervous system. We also have a peripheral nervous system. Peripheral just means, more on the edges. We refer to this sometimes as the PNS, the peripheral nervous system. It subdivides into the somatic nervous system, which we're not going to talk a lot about, but if you're an ENS major, you're going to be learning about that. What we're really interested in is the autonomic nervous system, the ANS, and we're interested in this because it's going to come back at least 3 more times this semester, where we're going to revisit this when we talk about things like emotion, stress, and health. We talk about things like psych disorders and treatment for psych disorders as well. The autonomic nervous system basically is this, guys. It's the system that is in charge of maintaining homeostasis. Autonomic basically means automatic. It does things like digesting your food, and it does these things automatically. Like if I said to you right now, I want you to really concentrate and digest your food. Go ahead. Work really hard on that for a second. It's just happening, right? It's got 2 subdivisions that work in opposing directions. When one is turned on, the other is necessarily turned off. The 2 subdivisions are sympathetic and parasympathetic. I'm going to give you a mnemonic device to keep them straight because the names are so darn similar. The sympathetic division is the F or F system. What do you think F or F means? Fight or flight, okay? So this is the part here that's involved with helping you to survive in an emergency, okay? If you're walking down the street and somebody jumps out from behind a truck, it scares the daylights out of you. You know what that feels like physically, right? Your heart starts racing. You start breathing really rapidly. Your mouth gets dry. Your palms might be sweaty. Your hands are probably shaking. All of that stuff is sympathetic division activation. And this system is really about utilizing whatever resources you have to fight or fly or get out of there, right? So if we get specific about this, these are the kinds of things that happen. Okay, the shock occurs, your pupils will dilate, they become bigger, your breathing becomes more rapid, your heart rate increases, you get increased blood flow to your muscles. Look at where the only downward arrow is. Digestion slows down. Okay? Let's imagine that you're swimming in the ocean and you see that caudal fin of the shark. And maybe you even hear like dun 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 dun, right? You're going to have a physiological reaction to that. This fight or flight system is going to kick in. It's going to include these things that activate everything other than digestion. And the joke that I use for this is, if you're going to become the shark's lunch, there's no point in digesting what you had for lunch, right? That's not really helpful. You get the increased blood flow to muscles. It includes squeezing in the blood from the periphery. So if you've ever had something really shock you or scare you, did you notice you had tingling in your fingers and toes? Or they might have felt cold? Anybody have ever had that? That's squeezing the blood in to the bigger muscles, right? So if you're about to get eaten by the shark, we don't want the blood in your fingers. He's probably going to bite off at least that much. Okay? So let's bring it into the bigger muscles. And I'm not suggesting that I have big muscles at all. But here is bigger. This is definitely bigger. But bring the blood in there. Don't leave it down in the foot because he's probably biting that off. Right? So we squeeze that stuff in. And whatever is necessary to survive, this is what we turn on here with the sympathetic division. What's the parasympathetic division do? Well, the way I'm going to try to help you guys remember this is parasympathetic division is the division of peace. Parasympathetic begins with P. Peace begins with P. Oh, that sounded like a clicker travesty in the back. What happens here with parasympathetic? The opposite of what goes on with sympathetic. So we constrict the pupils. The breathing slows down. The heart rate slows down. What's the only thing that becomes more active? The digestion. What time of year can you think of when you've probably got this going on? Thanksgiving. This is right after Thanksgiving dinner, right? That's my prototype for parasympathetic. Here it is. Slowing down the breathing. Barely could stay awake. My eyes are slamming shut. Heart rate is down. The Cowboys are losing again. It's all there. It's good. All that stuff is happening, right? So it's the opposite of all of this. This is the emergency system. This is the conserve resources system, okay? We're going to come back to this 2 more times. When we talk about emotion, we're going to talk about how emotions are connected with this. When we talk about psychological disorders, where do you think psychological disorders overlap with the autonomic nervous system? What kinds of disorders? Do you turn this stuff on too much? This kind of shaky. What does that feel like? Anxiety, right? Anxiety disorders, things like phobias and so forth, are really about turning on the sympathetic division when we probably don't want to. And in fact, when we parlay this into a discussion on treatment, the treatment is going to be for a phobia. We're going to teach you to do parasympathetic, calming, relaxation techniques rather than getting all worked up, right? That's where we're going to go with this. It's called systematic desensitization, and it's built on the way that your system is built. You cannot simultaneously be relaxed and really, really nervous. Try to do it. Can you? You can't, right? Once one is on, the other's off. And when the other thing is on, then the opposite thing is off. Okay? So we'll talk about that when we get to that segment. We'll also talk about it, like I said, on emotion, stress, and health. The health part of this is if you turn on the sympathetic division too much, what's this going to do to things like your lungs and your heart? Let's go with heart. If you keep cranking on the emergency system for your heart all the time, what's going to happen? It's going to be bad for your heart over time, isn't it? Right? And the book I'm going to mention in that section, I can't not talk about it. There's a book called, believe it or not, the title is Why Zebras Don't Get Ulcers. It's a great title, right? Why don't we get ulcers? Because what do? We worry about, what do we worry about? Like everything, like the stupidest stuff we worry about, right? When do zebras worry? When the lion is about to eat them. They're like, oh shit, turn on the system, I'm going to get eaten, I got to get out of here. Right? The zebra, think about it, zebras don't like lie there in the zebra house and be like, oh, I got like a big paper due Friday. Really worried about that, right? The zebra's like, that new zebra baby doesn't look very much like me. Like, I don't know what's going on there. I'm worried about that. No, right? Zebras just, like, they say, turn it on. Like, if you're about to get eaten and dead, then turn it on. But here's you and me, like, oh, my car made this weird noise this morning. I hope it starts later. Like, oh, it's going to be hot this week. Oh. Like, so what? Right? And we just worry, we turn this on way too much. I'm as guilty as anybody on this dimension for sure. All right, let's get into the final segment here, final 12 minutes or so. Now we zoom to the cellular level. What we're going to do here is say, like within the hippocampus, or whatever is your favorite part of the brain, what's going on at the cellular level? Well, our major focus and the star of the show is going to be the neuron. And the neurons help do communication within the central nervous system, the brain and spinal cord. So they receive, process, and or transmit information to other cells within the body. What I want to give you next on the final two bullet points on the left side here is a framework for keeping things straight. If you're taking the test next Tuesday and you get lost for a second and you're like, okay, it's a question about this and it's biology and it's something about neural communication, come back to this framework. The framework is communication here is electrical within and chemical between. If you let this be your anchor, you'll never get too far afield in what you're doing with the bio stuff. So what do we mean electrical within, chemical between? Electrical within is going to be about action potentials. I'm going to write APs. How many guys learned about action potential somewhere along the line? Okay, that's not enough, so we'll cover that on Thursday. And then it's chemical between, which has to do with the functioning of these things called neurotransmitters. That's what the NT stands for. How many guys have heard of neurotransmitters somewhere? It's actually a few more, okay? So electrical within, chemical between. Just keep that as your mantra. You're going to hear me say it like at least a dozen times between now and when we get done on Thursday, okay, the final class before the test. Electrical within, chemical between. So the neurons are going to be the lead actor or actress in this show, but we do have a supporting actor, actress in the form of the glia. They do a bunch of important things. So I don't want to give them short shrift, but we're going to mostly focus on neurons. The glia do about 5 different things I want to mention here. Number 1, they hold the neurons together. That's where the word comes from. The glia word is actually from the Greek word for glue. Why do they hold neurons together? Because it turns out, here's the deal, that the neurons, when they sit adjacent to each other, imagine these are 2 neurons, they don't actually butt up against each other and touch each other. There's a little gap that we're going to call on Thursday the synapse. And so they sort of float here, and what holds them in place, the surrounding structure is really this network of glial cells. The glia also help facilitate neural transmission. They help messages move through the central nervous system. I'm being purposely vague there because it's intro and I don't want to just overwhelm you. It already feels like you're drinking from a fire hose, so let's just say they help with that. The biopsych professor can add more. They play a role in cleanup. They remove damaged and dead neurons. If you have a stroke, if you have some kind of head injury and neurons die, they're involved with sort of flushing that stuff out and cleaning up. They're involved in keeping bad things from getting into the brain in the first place. So sort of playing the role of bouncer here, right? Your central nervous system bouncer is the glial cells, keeping out toxic substances, these teratogens, things that could get in there and cause you problems. And probably most importantly, I don't know why I have it last since it's most important, is the role that the glia play in building myelin sheaths. As we're going to learn in a minute, the myelin sheaths help to insulate this electrical within communication. Effectively, the myelin sheaths are like the plastic or rubber wrapped around the wires in an electrical cord. And they keep this electrical signal within the neuron from dissipating into the extracellular fluid. By the way, it's the myelin sheaths that actually break down when someone has multiple sclerosis. It's actually easy to remember because myelin sheaths and MS both begin with MS, right? So we get this breakdown in myelin sheaths in the context of multiple sclerosis. If you have insulating material that is breaking down, messages moving through that system are not going to go through as quickly or as easily as they otherwise would. And so you get the symptoms that you get in the context of MS, that it can include motor symptoms and some other stuff as well. All right, I want to do 2 slides in the last 8 minutes. One is a picture, and the other is basically the picture turned into words. And what we want to look at is the major parts of the neuron. There are 6 of them that I want you guys to be responsible for, and we're going to circle back to this at the beginning of our get-together online on Thursday. So, major parts of the neuron. We're going to begin with number 1 here, the dendrites. It says that dendrites receive messages. So, if this neuron that we're looking at is receiving a message from somewhere else, that message is coming in through the dendrites. All of these guys. See these thin, spindly, kind of branch-like structures here? A whole ton of dendrites. Structure number 2 is the cell body. What's the other word for cell body that you'll sometimes see? Soma. Yep. Cell body or soma. So the cell body's got the cytoplasm, it's got a nucleus and all that stuff that goes back to maybe 4th or 5th or 6th grade science. So the message comes in through the dendrites, moves through the cell body, and then it moves down the axon, which is going to be structure number 3 here, the axon. I got a mnemonic for that one for you too. The axon sends the message away from the cell body. So axon and away both begin with the letter A. Okay? So now we're moving away from the cell body, down the slide this way, and then it sort of pivots and goes to the right across the slide until we ultimately get to the endings here called the terminal buds in this figure from your textbook, figure 2.1. You'll hear these terminal structures called terminal buds. You'll hear them called terminal endings. You'll even hear these called terminal buttons. Okay? So a lot of structures here I just want to review with you guys before we wrap up for today. So terminal endings or terminal buttons is the term you'll hear. It's these guys here, right? All these are the terminal endings or terminal buttons there. Okay? So what's the sequence here? Again, I can use my hand as a model for the neuron. What are the 4 fingers here? The message is coming into the what? Dendrites. The palm of my hand is cell body. My thumb is axon. We've got 1 axon that carries a message away. And then if I had weird, creepy things hanging out the end of my thumb, those would be the terminal buttons. 2 more things I want to add here. Notice these green segments. Oh, that's coming all the way down. These are myelin sheaths. So this would be structure number 5, built by the glial cells. The myelin sheaths speed up this electrical communication within the neuron by effectively making it so that that message doesn't roll all the way through that axon. What it does is it actually jumps from node to node here. The myelin sheets make it that fast. Those unmyelinated sections are the final structure here. Those are called nodes of Ranvier. R-A-N-V-I-E-R. It's kind of small on the slide. I want you to mention one thing in your notes about nodes of Ranvier. We got 4 more minutes, so don't get all shuffly on me. It's at the nodes of Ranvier that we have these structures called ion channels. We're going to say more about them on Thursday, but here's the deal, guys. I said it's electrical within, right? Ions are electrically charged, right? The ion channels at those little orange spots where you can see the axon, those nodes of Ranvier are just unmyelinated sections of the axon where you have swinging doors. You know, like the doors that go into and out of the kitchen in the restaurant? That's what these are like. Okay, these swing open or close, and they'll let some ions that are outside come into the cell, and they'll let some ions that are inside the cell go out. That's what we're going to be talking about in our discussion on Thursday. So again, dendrite, cell body, axon, terminal endings. We've got the myelin sheaths insulating it. We've got these ion channels at those nodes of Ranvier. We're not done. Hold on. You don't need to write those 2 things down. I'm just going to say it again. Electrical within, that's action potential. Chemical between, that's neurotransmitters. Here's the sequence. This is a great test question. I can give you 4 different sequences and only one of them is right. I guess if you're trying to get a mnemonic here, it's DCAT, right? Dendrite, cell body, axon, terminal buttons, terminal endings, whatever you want to call that last piece. That's the action potential. That's the electrical within. So if I want to be explicit about this and connect this up to what we've got up above, here's what I do. This is the electrical within part of the story. And then what happens is when that electrical charge gets to the terminal endings, the neurotransmitters do the chemical between communication. And here's the deal. When neuron number 1 releases a neurotransmitter into the synapse, into that little gap between neuron number 1 and number 2, neuron number 1 releases a neurotransmitter. We're going to learn on Thursday that it goes across the synapse, basically drives over and binds to a receptor site on neuron number 2. And what happens is this really rapid cascading sequence of like dominoes falling over. Electrical within causes chemical release between, which starts electrical within number 2, which causes chemical between 2 and 3, which starts electrical within number 3. You get the idea, right? Very, very rapidly, on the order of milliseconds, that message is moving through. On Thursday, we'll circle back to this, and then we're going to zoom in and look at the communication between neuron number 1 and neuron number 2.